Hello, in this video lecture, I'm going to talk about basic cloning steps. So I'm going to talk only about the plasmid vectors that you use to clone a piece of DNA. I will not talk about the phage vectors or the cosmid vectors or YAG vectors whatsoever, because it will make the thing complex. So let us talk about how you can use your plasmid vector as a cloning vector to clone a piece of DNA that is a gene of interest. So how do you isolate the gene? That is the different question. Suppose you have isolated a gene. It may be a PCR product, or you have screened out that particular gene by Southern blotting or whatever method. So if it is Southern blotting, there will be many fragments that you need to clone and make a library. But if you have done a PCR, it will be a single kind of a DNA insert. So in both cases, the thing will be same, that you are going to incorporate a DNA molecule within your vector, that is the plasmid vector. So now you have to take your plasmid, you have to select a particular plasmid in which you are going to clone. Say that plasmid is PBR322, or it may be PUC vectors like PUC8, PUC9, or PUC18 or PUC19. So whatever the case may be, your vector has restriction sites. So you will cleave your vector with a particular restriction endonuclease. So this time your endonuclease is EcoR1, just for our understanding. So you are using EcoR1 to linearize your circular plasmid DNA molecule like this. So it will generate two sticky ends, okay, because EcoR1 introduces staggered cut. Now you will use the same enzyme to produce your inserts. So your inserts are either it is a PCR product, so it will have two linkers of EcoR1 attached to it on the ends, or if it is a DNA, a genomic DNA, then you have to digest it with EcoR1, so that it also produces sticky ends, same sticky ends that are present within your vector, so that they will hybridize and you can ligate them. So now after this process is completed, you have to remove the phosphate, 5' phosphate, because these overhangs, the 4 nucleotide overhangs are the 5' overhangs. So you have to remove this 5' phosphate from the vector. Otherwise, if you do not remove it and you allow to ligate, if you put in the ligation reaction, it will reseal. It will religate itself without incorporating the DNA of interest. That is called insert. Without incorporating the insert, it will self-ligate within itself. So most of the molecule will be self-ligated. And by chance, if some molecule enters within that, the number of recombinants will be very, very less. So to avoid that, you have to remove this phosphate from the 5' end of your vector. So now this vector will not be able to religate or reseal itself. However, there are some reactions which are some vectors which do not get their phosphates removed, because no reaction is 100% complete. So some of the molecules will be left, and those will come in during your ligation process, in your ligation mix. So, but 90% of the cases, your vector will have no phosphate group at the 5' end. So they will not get resealed. They will only get sealed if they incorporate their insert. So now you are going to add your vector and your insert within a microfuse tube in a very small volume, maybe 10 microliter, 20 microliter, or maybe sometimes 50 microliter. So within that small volume, you have to take into consideration the ratio, the insert to vector ratio. The insert must be higher at concentration, so that the chance of getting incorporated in the vector will be more. So when you put them together and you add ligase enzyme, you have to incubate this reaction mix at a low temperature, 16 degrees centigrade. Since it is EcoR1, you have to incubate even at lower temperature, that is 12 degrees centigrade, because the EcoR1, it produces an overhang which has AT. So A and T, they hybridize at a much lower temperature because there are two hydrogen bonds between A and T. That's why when you are using EcoR1, you have to decrease the temperature further. And when you are using other enzymes like BAMH1, SAL1, you can do it at 16 degrees centigrade. So after overnight ligation, you have to take the samples, the ligation mix, and you have to transfer it into a host cell. And that host cell is an E. coli host cell. So at the same time, when you were putting the ligation reaction yester evening, you had inoculated a bacterial culture, E. coli bacterial culture. There are several strains of that, like the E. coli XL1 blue, DH5 alpha, or JM109, whatever you are using. Those are all AMP negative strains. So you have inoculated a culture, maybe 3 ml or 5 ml culture. After overnight growth, you prepare the culture, you harvest the culture, and you treat it with maybe calcium chloride, 0.1 molar calcium chloride treatment about 1 hour in ice will make these E. coli cells competent to take the outside DNA. So your cells are ready on one hand, your ligation mix is ready on the other hand. Now you are going to add the ligation mix within the cells, and you will use heat chill method to make your cells porous so that the DNA present outside will get internalized. So what is done is that you have to treat your cells at 42 degrees centigrade for 90 seconds only. And immediately you have to chill it. So in this heat and chill process, what happens? Your cells become transiently porous and they acquire the outside DNA. They take up the outside DNA within them. The cells get a shock. To recover them from that shock, you have to give a little bit what we call revival media so that the cells get revived for 1 hour or so. This media is a little bit rich media. So after that, you have to plate these cells that have acquired the recombinant DNA in petri dish in agar plates containing AMP within them. So there will be 3 types of cells in a ligation mix. The first one is the cells which have not been transformed. The second one, they have acquired the self-ligated, resealed, empty vectors, and the third one that you require, those have acquired the recombinant DNA molecule. So all these 3 cells are present in your microfuse tubes. About 200 microliter cells are there. Now you have revived it after adding into media, and now you are going to plate that. You are going to plate that on agar plates. In this time, you will add ampicillin in the plate. So that cells which have not acquired any transformed, that those have not been transformed, will get killed, will be removed over here. So what you are going to get are all the cells, they have been transformed. But some of the cells are recombinant and the others are non-recombinant. They have acquired only the self-ligated vector. So the screening process is the second step. So now you will go for screening. If it is PBr322, you will go for replica plating. This process is called, the way it is screened is called insertional mutagenesis. The insert that you have incorporated is present within a particular gene, that is TET-resistant gene. So your insert has gone inside that TET-resistant gene. So the TET gene is now inactivated. So the recombinant DNA will have this TET gene inactivated. So the cells, those receive the recombinant DNA, will be TET sensitive, and those which have received the empty vectors will be TET resistant. So now if you plate if you replica plate for using this as your mother plate, master plate, you make 2 replica plates. In one of the plates you put tetracycline, in other you put ampicillin, just like your mother plate. And you just, in replica plating, what we do, we produce the colony positions same by putting inverting the petri dish over a velvet coated wooden log that is sterile, so that the colonies get stick to the velvet, and again you lift it and you put the other plates over there in the same position, so that the colonies which have stick to the velvet plates will get again stick to the media and they will grow over there. So in this way you will produce the replica of this master plate in the 2 plates. So after this you will get these 2 plates. In one of the plates, all the colonies that are present in the master plate will grow, that is the AMP positive plate, and the other plate, that is TET resistant, the TET containing plate, will have some of the colonies missing. And these are the colonies, those are recombinant. And you can lift these colonies from the other plate by just comparing it, the position wherever it is absent, there is a colony over there in your daughter plate or master plate whatsoever. So you can, you can pick up the colony from there. That is the recombinant colony. This is how. This process of screening is called negative screening because you are screening something which is absent. So this type of screening process is called, or selection process in bacteriology is called negative screening. So this is how you clone your DNA and you get your recombinant DNA screened if you are using PBR322. So if you are using PUC vector or PBS-SK plus vector or similar kind of vectors, those have ligase gene as the marker gene for selection, recombinant selection, your multiple cloning sites are within this ligase gene. So when you have inserted your DNA gene of interest, it has inserted and inactivated the ligase gene. The ligase gene or ligase gene, it produces beta-galactosidase. And this beta-galactosidase, as you know, this is the counterpart of that lac operon. We are not going to use here lactose. We are going to, instead of using lactose, we are going to use two different kind of chemicals. One is the inducer, that is IPTG, isopropyl thiogalactoside, and the other is X-gal. This X-gal is the substrate that will get converted into a blue colored product by this enzyme beta-galactosidase. So what will happen in the Petri dish when you are going to plate after you have incorporated or transformed your E. coli cell with the ligase mix, when you are going to plate on a Petri dish, in the Petri dish you have to have ampicillin first of all, and then that plate should also have X-gal and IPTG. Now, because of the presence of ampicillin, cells which have not acquired any vector or plasmid will be killed. But those cells which have acquired the plasmid, the empty vector as well as the recombinant plate, they can be distinguished by this plate as those cells which have acquired the self-ligated vector will be blue because their ligase gene is intact and they will produce beta-galactosidase, and that beta-galactosidase will convert the X-gal into a blue-colored product. These colonies will be blue. And the recombinant colonies, their ligase gene has been disrupted because of the insertion of the foreign DNA, that your gene of interest, and so the ligase gene is inactivated. It will not produce beta-galactosidase, and it will not be able to convert X-gal into a blue-colored product, so the colonies will be white. So you pick up the white colonies, these are your recombinant clones. So this was how basic cloning is done using these plasmid vectors. But one thing remains to be said over here. When you are putting the insert and ligating with the vector, you will see that there are two sites where 5' phosphate of the insert and 3' hydroxyl group of the vector, they come side by side, and your ligase will seal it, will form a covalent bond over there. But on the other end, the other nicked area, where your vector has a 3' hydroxyl group and the plasmid has a 5' hydroxyl group because you have removed the phosphate, that end will remain unsealed. And you are going to transform this particular DNA there are two nicks that are unsealed into the E. coli cell. After going into the E. coli cell has its own kinase and ligase, with the help of which it will incorporate a phosphate at the 5' end, and it will seal the gap also there. So the whole process is going to be completed after the transformation. So this is what very important, that place has to be significantly noticed. Thank you very much for watching this lecture.